Hello viewers, welcome back to this particular second part of the cardiac cycle. I, SANKRANTI Devi, will now discuss about the electrocardiogram and also the factors which are responsible for the changes in the ECG. The Wiggers diagram will also be discussed here, and also the factors which are responsible for the change of the cardiac output, and also the all total summary of the two presentations. Talking about the ECG machine, that is electrocardiogram machine. If we go to 1903, we can see that a famous scientist, Willem Einthoven, a Dutch doctor and also a physiologist, he invented the first practical electrocardiogram and received a Nobel Prize for medicine in 1924. Here is the beautiful picture of the electrocardiogram which was invented in 1903. Now if we analyze the electrocardiogram, we can see here it's a beautiful picture of the normal ECG of a human being. Here we can see different parts, that is the P, a QRS peak and a T peak. If we discuss about this, an electrocardiogram or an EKG or ECG is a test that measures the electrical activity of the heartbeat. With each beat, an electrical impulse or wave travels through the heart, which causes the muscles to squeeze and pump the blood from the heart. In the figure, we can see the right and the left atria make the first wave, which is known as the P wave. This P wave symbolizes that there is a contraction in the atria. It follows a flat line when the electrical impulse goes to the bottom chamber, and this part is known as PR segment. The right and the left ventricle makes the next wave, which is known as the QRS complex. The QRS complex demonstrates that there is contraction of the ventricles. The final wave, that is the T wave. Before reaching the T wave, we can see another flat line where the electrical impulse again moves from the bottom chambers to the upwards. This is represented by the ST segment. The T wave is the electrical recovery or the return to a resting state for the ventricles, that is the repolarization of the ventricles. Here we can also see two intervals, that is the PR interval and the QT interval. So this diagram basically gives us the explanation what happens when a heartbeat occurs, the electrical activity that occurs during each heartbeat. Here is again a very beautiful diagram in which the excitation of heart is related to the ECG. The first phase, where we have already seen the SA node originates the impulse. Here the P wave occurs, that is the atrial depolarization. Again, the PQ or the PR segment which we have discussed, it is the conduction of impulse through the AV node and the AV bundle. That means the atria, which is purplish in color, demonstrate that the impulse has traveled whole through the atria and the atria contract. Moving on to the third part, in which we can see only a small part of the heart that is in between the ventricles, it is purplish in color, which is represented by the Q, starting of the Q wave. Now moving on to the fourth part of the diagram, that is where the impulse is moving from the middle of the ventricles towards the muscles of the heart. in which the R wave is generating. Afterwards, when the ventricles already starts to contract, bit by bit, the impulse travels through the ventricles and it results in the formation of the S wave. Thus, the QRS wave or the QRS complex is complete. This is represented by the ventricular contraction. In the sixth part of the diagram, we can see that the ventricles have contracted. Again, when the repolarization starts, we can see the formation of the T wave. And in the last diagram, which is the end of the ECG, we can see that there is no impulse, no contraction and no relaxation. That is, a flat line is shown there. This whole diagram shows how the heart is excited, and this excited, how it is related to the electrocardiogram. That means the electrical events which occurs in the cardiac cycle and its representation with the help of the electrocardiogram. Also a similar diagram as we have seen it before. During the atrial depolarization, there is the activation of the atria. During the ventricular depolarization, that is the QRS complex, there is activation of the ventricles, and during the ventricular repolarization, it is the recovery wave. The normal heartbeat, fast heartbeat, slow heartbeat and irregular heartbeat are depicted here, and we can beautifully differentiate between them. In the normal heartbeat and the fast heartbeat, we can see the difference that the PQRST parts of the ECG is occurring more rapidly, whereas in the slow heartbeat it has much time interval. And in the irregular heartbeat, we can see that there is no definite rhythm which is followed. This is the beautiful Wiggers diagram which contains the overall theory of the cardiac cycle, which includes all the volume, pressure and ECG graph in total. It is beautifully depicted when the heart sounds occur, when the volume goes up, when the volume goes down, when there is pressure increase, when there is pressure decrease, and also the electrocardiogram. The atrial systole and different phases of the cardiac cycle is related to the volume, pressure and the ECG of this gram. Looking into this beautiful diagram, we can easily relate the events that we have studied. Now talking about another topic, which is known as the cardiac output and reserve. Cardiac output is the volume of blood which is being pumped out by the heart, particularly the left and right ventricles, in the time interval of 1 minute. It is generally calculated by the formula, which is cardiac output equals stroke volume into heart rate. As we have already discussed, stroke volume is the amount of blood given out by the left ventricle in one contraction, and the heart rate is the number of times that a heart beats per minute. And in a resting person, the cardiac output is about 5.25 milliliter per minute approximately. Now talking about the cardiac reserve, that means it is a difference between the resting and the maximal cardiac output, the amount of blood that is reserved per minute of the cardiac cycle. There are generally certain factors which affect the cardiac output, and these factors can be studied by this beautiful diagram. Cardiac output is generally, we have seen, is a function of heart rate and the stroke volume. If we see the heart rate, we can observe that it is determined by the rate of depolarization in the autorhythmic cells. which if decreases, it is due to the parasympathetic innervation. And the sympathetic innervation and epinephrine hormone causes the increment of the heart rate. Now if we see the stroke volume, here we can see that it is determined by the force of contraction in the ventricular myocardium, and it generally influenced by two parts, which is known as contractility and end diastolic volume. The sympathetic innervation or the epinephrine generally when increases the contractility, it increases the stroke volume. The end diastolic volume, it varies with the venous return. And when there is venous constriction due to epinephrine and sympathetic innervation, the venous return in return increases. The venous return is aided by two things. One is the skeletal muscle pump and the respiratory pump. So this diagram basically tells us how different factors are affecting the cardiac output. Now we can see a simple law which was given by Frank-starling. It defines that within the physiological limits, the greater the heart muscle is stretched during filling, the greater will be the force of contraction and there is greater quantity of blood pumped into the aorta. From this beautiful diagram, we can see that if a graph is plotted with the force by stretch, that is force which is indicated by the stroke volume and the stretch which is indicated by the ventricular end diastolic volume, we can see if the force and stretch both are increased, then there is more quantity of blood pumped into the aorta. As we can see, the graph reaches almost about 380, which tells us that when the heart muscle is stretched to a more level, filling is more, contraction is more, and thus the quantity of blood pump is also more. We have a normal resting values which are 135 and 70, which is there is the force is almost 70, that means the stroke volume ml, and the stretch which is indicated by the ventricular diastole volume which is 135. In these two heart diagrams we can see two phases. One is the preload and one is the afterload. Preload means before loading of blood into the ventricles, the stage, and afterload means when the blood is pumped out of the ventricles. As we can see that there is more pressure into the ventricles during the preload and there is less pressure in the ventricles during the afterload. Now moving on to the reflex control of the heart rate, we can observe that the cardiovascular center of control, which is present in the medulla oblongata, has two major effects. That is, when the norepinephrine, that is the sympathetic neurons, acts on the beta-1 receptor cells of the autorhythmic cells, there is sodium and calcium influx, and as a result of which there is an increase in the rate of depolarization, thus increasing the heart rate. But if we see the parasympathetic neurons, that is the acetylcholine, the parasympathetic neurons causes the muscarinic receptors of the autorhythmicity cells to activate, which results in potassium efflux and calcium influx. This hyperpolarizes the cell and the rate of depolarization decreases, which results in the decrease of the heart rate. In the key we can see that the square or the kite part of the diagram is the integrating center, and the purplish figures represents the efferent path. The bluish parts represents the effector and the greenish parts represents the response of the tissue. From this flowchart we can know the reflex control of the heart rate, how it is stimulated or affected by the sympathetic and the parasympathetic neurons. Moving on to the summary part of the whole cardiac cycle, if we summarize the points, we can see that the starting point is where the SA node generates the impulse and all the four chambers are relaxed and the ventricles are partially filled with blood. During the atrial systole, the atrial contract and complete filling of the ventricles occurs. The atrial systole lasts for 100 milliseconds. After the completion of the atrial systole, the atrial diastole begins and continues until the start of the next cardiac cycle. As the atrial systole ends, the ventricular systole begins, and this period, which lasts for 270 milliseconds, can be divided into generally two phases. The ventricular systole first phase is the contraction phase, which pushes the atrioventricular valves closed but does not create enough pressure to open the semilunar valves, and this kind of situation is known as isovolumetric contraction. The next phase, which is the second phase, is where the ventricular pressure rises and exceeds the pressure in the arteries. The semilunar valves open and the blood is forced out of the ventricle, which is known as ventricular ejection. Now moving on to the ventricular diastole, in the early phase, the ventricles are relaxed and the pressure in them drops. Blood flows back against the cusps of the semilunar valves and forces them to close. The second phase, where the blood flows into the relaxed atria but the atrioventricular valves remains closed. This is known as the period of isovolumetric relaxation. And in the eighth phase, where the ventricular diastole occurs, in the late phase, that all the chambers are relaxed here and the ventricles fill passively to roughly about 70% of their volume. The ventricular diastole lasts for about 530 milliseconds. The 430 milliseconds remaining in this cardiac cycle plus the first 100 milliseconds of the next. Throughout the rest of this cardiac cycle, the filling occurs passively and both the atria and the ventricles are relaxed. And the next cardiac cycle begins with atrial systole and the completion of ventricular filling. So as we have come to the end of our presentation, these are some of the suggested readings, that means the books I have followed, and these are some of the research papers that I have studied for making this presentation. Thank you so much viewers for your patience hearing. Thank you.